Sathyabama Institute of Science and Technology.
Good morning to everyone. I am Jancy Mary, Assistant Professor, Department of Biotechnology, Sathyabama Institute of Science and Technology, Deemed University. Today we will discuss about the topic absorption of carbohydrate. So digestion of carbohydrate, it occurs in mouth, stomach and small intestine. This then it can be absorbed by three mechanisms. One is passive diffusion, another one is facilitated diffusion, next is active transport. So first one is facilitated transport. This facilitated transport, all the monosaccharide are absorbed easily in facilitated diffusion method. But active transport, it requires the sodium dependent glucose transporter, that is also called as SGLT. So it is called as co-transporter because the sodium and glucose at separate sites and transport them into the cells. So this is absorption of carbohydrate by sodium potassium pump. With the help of the energy called ATP, it is absorbed to intestine. So these are the some of the features of that three mechanisms. One is passive diffusion, next is facilitated diffusion, next is active transport. In this, the concentration gradient is first two, passive and facilitated diffusion, that is high to low. But in active transport, it is low to high. The energy expenditure is focused on active transport in the form of ATP. Carrier protein, it is not required in passive diffusion, but in facilitated and active transport, it requires the carrier protein or transporters. The speed is slowest in passive diffusion. The active transport is more fast. So the glucose transporter are of two types. One is sodium dependent transporter, another one is sodium independent transporter. So sodium dependent transporter is also called as SGLT. Sodium independent transporter is also called as GLUT. So this one is sodium dependent transporter. In this sodium dependent transporter, it is also called as co-transporter. It transports both glucose and sodium as well as this sodium is transported across the cell membrane in concentration gradient, and the glucose also against concentration gradient. In this process, it requires ATP in the form of energy. The glucose and galactose are absorbed in sodium dependent process. So this is SGLT form, that is SGLT 1 or 2. SGLT 1 is present in intestinal cells, SGLT 2 is present in renal tubular cells. Both the cells, it requires sodium potassium pump reaction. The three reasons for the expulsion of sodium. First one is this sodium, it leads to osmolysis. Next is sodium, it maintains the gradient, that is downward gradient. Next is sodium, it inhibits many enzymes. So this is solvent drag absorption. First, the sodium releases the energy, then this sodium expelled from sodium potassium pump, then 3 sodiums are expelled and 2 potassiums are expelled. The sodium is removed from some spaces, that is intracellular spaces. Then water carries the dissolved glucose. This type is called as solvent drag absorption. Next is the clinical significance. Mainly SGLT. The clinical significance of SGLT1 is galactose is not absorbed properly. That is called as SGLT1. But SGLT2, that term is called as glycosuria. That means the glucose is more excreted in urine. Next is sodium independent transporter. It is used for facilitated transport. These transporters are numbered from 1 to 14 GLUT. This GLUT2 is mainly present in intestinal epithelial cells. GLUT5 are present in luminal surface. So GLUT2 and GLUT5 of this 14, this 2 is mainly involved. GLUT2 is present in blood. GLUT5 is present in lumen. By sodium potassium pump, the junction, it is absorbed. The purpose of GLUT2 and 5, the GLUT5 is mainly transported to fructose by facilitated diffusion. The process is very fast compared to GLUT2. This GLUT2, it transport the absorbed glucose to blood. The transporter present is slow and it does not require any energy. Next is, what are the factors affecting absorption of monosaccharide? The absorption is faster, the absorption is decreased when the inflammation injury is formed. The thyroid hormone is increased, the rate of absorption is also increased. The mineralocorticoid, that is aldosterone hormone, is increased, the rate of absorption is increased. Of this, the absorption required some of the vitamins like vitamin B12, pantothenic acid, vitamin B6 and folic acid. With this advancing, the rate of absorption is declines. The insulin does not play any role in absorption of this carbohydrate. Uptake of glucose in peripheral cells, the main mechanism is facilitated diffusion. There are 7 important glucose transporters. They have been numbered from 1 to 7, GLUT. So first, GLUT1, it is present in all cells, it is sodium independent. The cancer cells mainly express GLUT1. GLUT2, it is present in intestine. This releases insulin by movement of glucose. It is, the clinical significance is diabetes mellitus. GLUT3, it is present in brain cells. The cancer cells highly express this GLUT. GLUT4, it is present in adipose tissue and skeletal muscles. The only transporter involved is insulin. GLUT6, it is non-functional transporter. GLUT7, it is present in endoplasmic reticulum. Mainly it transport glucose across endoplasmic reticulum. SGLT1, that is present in kidney and intestine for the absorption of glucose. In some deficient, the glucose is not absorbed. SGLT2, it is present in kidney. It is involved for the reabsorption of glucose. If some deficient, the glucose is not reabsorbed. These are main 7 glucose transporters, that is glucose 1 to 7. The glucose, that is GLUT1, it is present in brain, kidney, placenta and RBC. These are all involved for the uptake and release of glucose. GLUT2 is present in liver, pancreatic cell. GLUT3 is present in brain and kidney. GLUT4 is present in heart, skeletal muscle. GLUT5 is present in small intestine. Thank you.